Thanks a lot for the invitation and the introduction. So yeah, what I was planning today was to speak a bit of my recent transition to wake research. So Xavier has explained. I mean, the first part of my career, I investigated how the sleeping brain is processing external information, and now I'm interested in wakefulness and the fluctuation of our ability to pay attention to our environment while we are awake. So it's a program of research I developed in the past 3 years at Monash University, but now I'm continuing it at the Paris Brain Institute, where I'm located right now. And just to give you an idea of, how all this research program is integrated with itself. So I will repeat a bit of my biography, but it's relevant for the talk, and I will be very brief. But something I had the chance to do very early was to go to the lab of Giulio Tononi and Cara Chielli in the US for my master, where I had the opportunity to study sleep physiology in implanted patients, which is what's really great as a student, but also in terms of discovery. It was a unique opportunity to record sleeping humans at various scales, from single neurons to the whole brain. And what we discovered with this research was that the sleep was not the global phenomenon that is often described in textbook, but something more locally regulated. And you can have different parts of your brain in different vigilant states during the night. So during my PhD, I studied the consequences of that for cognition, because if different parts of your brain can be in different vigilant states, maybe that can allow you to process some information while you're sleeping. You can imagine that part of your brain will wake up to an external stimulation while the rest is still asleep, and that will allow you to process that piece of information. So it's something I investigated with Sid Quider, but also Mathieu, who is in this virtual room. And we showed that you can do many things while you're asleep. And these many things seems sometimes to correlate with some shallowing of sleep depth locally. But after this PhD and master, I wanted to take a break. So with my wife, we went for a 1-year trip around the world, and I was trying to figure out what will be my next research interest. And again, this is relevant for this talk because at some point during this trip, pretty much here in the middle of Patagonia, I flipped the car. We were traveling by car, which was not a great thing to do. Cars are not supposed to land on their roof, as you know. And we got down from the locals, so obviously no one was injured. It was a slow speed event, fortunately for us. But we had some help for local, and the immediate reaction was for them to ask, how did that happen? And they had different hypotheses. So either they asked whether I fell asleep, which was not the case. They had whether we had some encounters with vicuñas, which are the local llamas. This was not the case. And I had big difficulty in admitting that it was just thinking about something else at the time of losing control of the car. And more specifically, I was thinking about Birmania. Don't ask me why I was thinking about Birmania, but I was thinking about Birmania. And I thought, how come I can put myself in such a bad situation while being, I had the impression of being very much awake. I was actually a bit bored and thinking about something completely different. So how I could be so disconnected from my environment while having no problem of vigilance whatsoever. And it was through this accident that I thought, oh, maybe that would be interesting to understand why sometimes our attention is completely lapsing. So we moved to Australia, and I started working on this topic with Joel Pearson and now Tutia. And in particular with this idea of trying to understand the interconnection between fluctuation of vigilance and fluctuation of attention. And sometimes you can have the impression you're perfectly awake and you're not sleepy at all, but that doesn't mean that part of your brain is not feeling sleepy. And this is because this breakdown of the dichotomy between sleep and wake is actually not only observed only in sleep. So this notion of local sleep is not, it's not only local wake within your night, but you have parts of your brain waking up. This is something you can also notice during the day. So in a typical sleep, what will happen to your brain activity is that the EEG will slow down, and your brain activity will be dominated by high amplitude hypersynchronized events, which are called slow waves. And these slow waves at the neuronal level, they are associated with a form of neuronal bistability. So your neurons are very synchronized, they fire together, they get silent together. And that is the supposed mechanism why you're losing consciousness in the deepest part of dreamless sleep. When you're awake, the activity is completely different. You have desynchronized oscillation of low amplitude, fast rhythms, and your neurons are firing in an asynchronous way. So what is interesting to see in this two extreme is all the things in between. And in particular, in wakefulness, if you get people tired, what you will see is an emergence in the background of wake activity of local markers of sleep. So something that will look like a small sleep slow wave occurring locally in the brain while the rest of the brain is globally awake. And what I'm trying to understand is how frequently these local sleep events occur and what are their impact on the functioning of the brain. So these local sleep events, they have been first characterized in rodents using acute sleep deprivation. So you keep rodents awake for long periods of time. So it's a work done by Władysław Wiazowski when he was in the group of Giulio Tononi. And what they show is that if you sleep deprive rats for long enough, you will see appearing on the LFP, so inside the brain of the rats, but also in the single unit recordings, things that looks a bit like this non-REM slow wave. So a slowing down of brain oscillations associated with a decrease of the firing of neurons. These events can be rather global, so that you can see them across multiple brain regions, or on the contrary, they can be local. But what is very interesting is that they seem to index well sleep pressure. So they increase. So the more you keep a rat sleep deprived, so the more you will start observing this slow wave in the neural recordings. And the same slow wave decreases the more you sleep. So they increase the more you stay awake, they decrease the more you sleep, so they're almost regulated. And when they occur in wakefulness, they seem to predict behavioral errors on the task. So in this paper, the rodents were trained to do a task where they had to reach a food pellet. So it's a task involving the frontal cortices to decide when or not to reach for the food pellet. And what they observed is that before a miss, they had an increased probability of observing this sleep intrusion in the brain region involved in the task. So you can see that if you separate hits and misses, you can see more sleep intrusion, more slow wave in frontal cortices for the misses compared to the hits. But you don't see that relationship in parietal cortices, which are not involved in the task. So this was thought as a compelling argument to say that not only this sleep intrusion index tiredness, but they could also explain why we are making mistakes when we are feeling tired. So a similar approach has been replicated in humans. So if you sleep deprive humans for a very long period of time, so here 24 hours without sleeping, you can see that the scalp EEG is displaying more and more theta power So there, you can observe this slowing down of the brain oscillations. So it's work done in particular by Giulio Bernardi, Bessie Hung, and Simone Sara, so still in the Autonomy lab. What they showed as well is that you have some region-specific effects. So if you're asking a participant to do the same task for 24 hours straight, so it's a quite challenging protocol, you can see that the regions that are involved in these tasks will show more and more of this theta wave. So reading tasks will increase the theta wave over frontal cortices, while your driving tasks will increase theta wave over motor cortices. And what is interesting as well is that this increase in theta wave is related to behavioral errors, also in a region-specific way. So the region that are showing a relationship between increasing theta power and increasing errors corresponds to the regions that are involved in the task of interest. So this piece of work is very interesting because it shows that you don't need necessarily to stick up electrodes inside the brain to record this form of sleep intrusion. You can also use scalp EEG to detect when parts of the brain, specific parts of the brain, are getting tired. So with Gio Tononi, Zach Fried, and Yuval Nir, we set out to try to understand a bit better what was occurring when we get tired. And to do that, we use this ability of looking at activity at different levels of recording in humans performing a task. So in this case, we use a psychomotor vigilance task, which is a basic task that is done in sleep labs to measure vigilance in humans. It's a task where you're presented with images. So the classical one is just a red dot. We use faces and places because we were recording in the medial temporal lobe, so to drive well the neurons we were recording. But the typical task is very easy. You have stimuli that will suddenly appear on your screen, and you just have to press a response button as fast as possible. And obviously, when you get tired, sometimes you get slower or you miss the stimulus entirely. So the interesting part of this data as well is that since we're recording epileptic patients, these patients will actually undergo a procedure of sleep deprivation as part of their clinical routine. Because to be able to localize the epileptic foci, sometimes these patients go through protocol of sleep deprivation, which wouldn't be possible to do for only a research purpose. So the fact that they do that as part of their clinical routine allowed us to be able to record data in intracranial data in sleep deprived humans. And what you observe when you sleep deprive these participants, of course, is what I was telling you. So a general shift of the distribution of reaction time and an explosion of the reaction time that are particularly slow. So what we decided was to compare for each session the reaction times that were the fastest with the reaction times that was the slowest, and to see what are the differences in the neural recordings between these fast reaction times and these lapses. And what you can see that already at the neuronal level, you can see a slowing down of the response of individual neurons when people get tired, as if the behavioral lapses that you observe through reaction times were somewhat a consequence of neuronal lapses. So the fact that neurons themselves are getting slower to respond to visual stimuli, which can be seen by an increase in the latency of the response to these neurons with the images, and a decrease in the magnitude of that response. What was also very interesting in this data is that we didn't have only effects on the neuronal activity, but we could also observe effects in terms of oscillation. And in particular, we could observe that in slow trials, in lapses, we had an increase in slow theta power. So in these slow oscillations that are typical of sleep intrusion, as I was mentioning before. So in fact, what you can see that what we have in this slow oscillation is the slow frequency range is less of a desynchronization. So when you receive a stimuli, you will depart from the resting state. And what happens when you get tired is that departing from the resting state is more difficult, potentially because you have higher power in the baseline. So it's more difficult to engage in the task. And that difficulty in engaging in the task might be associated with this form of neuronal lapses I was mentioning before. So it seemed that from there on, in the context of sleep deprivation, we have a very coherent picture from animal recording to intracranial recording in humans to non-invasive recording in humans. If you sleep deprive an individual, their activity, their brain activity, start looking more like something like sleep, actually. That will delay your ability to respond to external stimuli, and that could explain why sometimes you're missing information from your environment. What I wanted to understand is whether this was the case as well outside sleep deprivation. So if you take a normal person in a normal day, will you still see that this intrusion, these slow waves that are typical of sleep in wakefulness, and are there a good index of how tired you are during your day? So what we did to do this, and it's a work that has been recently accepted in Nature Communications. So you will hopefully be able to read it soon as we have paid the huge bill. So what we did was to take normal people, have them doing a boring task and recording their EEG while they are doing that. So I will dive a bit more in the details of the protocol later, but I wanted to first show how the brain recordings we get, so the scalp EEG we get through this protocol, can be used to detect this slow wave. And what we obtain is waves that are pretty similar to the one you will observe in sleep. So they are not identical. They have some faster component, but you can detect this slow wave during wakefulness, and they are quite high amplitude, even by definition. They are not as high as what you would observe in sleep, because otherwise people will be asleep. But we have these waves. They are slow in frequency. They are high amplitude. They're mostly frontal in their distribution, so they share a lot of the properties of sleep slow wave. And they seem to index well both objective and subjective indexes of fatigue. So they increase, well, they're negatively correlated with pupil size, and negatively correlated with vigilance ratings. So it seems we have here a marker that is easy to detect non-invasively, because you just need scalp EEG, and this marker seems to be sensitive to how tired you are. So what we're interested in is whether these markers that we detect, again, in people that are not sleep deprived, so people that will be very close to you and me right now, we were interested to know whether these markers will be able to predict not only their behavior, so how they behave in the task, but also their phenomenology, so what's going on in their minds. Because these striking results that although we think we're very smart and in control of ourselves, we have a very hard time maintaining our attention to boring tasks. And there's this oft-quoted number that we spend half of our waking life mind wandering, so thinking about things we're not supposed to be focused on. So we wanted to see whether these markers of sleep could explain both not only the behavioral aspect of lapses, but also the subjective experience of people when they are asked to do a boring task. So in terms of boring tasks, I mean, there's no shortage of choice in cognitive sciences in terms of boring tasks. I think we, as a field, we have been master at making boring tasks. But But there is one task in particular that has been used a lot in the mind wandering literature, and it's the SRIT, which stands for the Sustained Attention to Response task. So it's a task where you have a string of stimuli coming in, and you have to press a response button for most of the stimuli. There's just one target stimuli for which you have to hold your response. So in the classical SRT, it's a digit SRT where you have digits appearing in random order, and you have to hold your response for the digit number 3. We made also a modified version of this using phases, where just to break down a bit the monotony of the task. So it was a block design. In some blocks, they had to do the digit SRT. In some other blocks, they had to do the phase SRT, where they had to respond for all the neutral phases and hold their response for the smiling phase. So it's a fairly boring task. I mean, the instructions are very simple, and all you need to do is to pay attention to what's in front of your eyes. What typically happens in the SRT is, since you have many more go responses than no-go responses, very often you get carried away and you press the button for the no-go response. So it's a task where you do a lot of false alarms. What we did during this task, that was fairly long. It ran for almost 1.5 hours. So we interrupted people every minute or so and asked them in which state of mind they were, whether they were focusing on the task, which we call task focus or on, whether they were not thinking about anything, so whether their mind was blank in a way. They couldn't really tell us if they were thinking of the task or something else. They had the impression that their mind was empty. So we called that mind blanking. And then we asked them to report when they were thinking about something else than the task, which we called mind wandering. Something maybe important to note is that in lots of mind wandering papers, you have actually a conflation of mind blanking and mind wandering, because the contrast is between being on and being off. So here we we divided the two, and I think it's important to separate these two types of attentional lapses. And it's by no means a perfect way of doing this. I mean, you have also subdivision, and we had actually other question about subdivision of mind wandering. So whether you're thinking about something personal or something in your environment, but then if you break out too much, you just don't have enough data to do your analysis, unfortunately. So that's the results of these mind probes during the task, and what you can see that in both the digits and the phase SRT. So I will always show the two tasks separately, although they were done in the same participants. What you can see that we replicate this very famous finding that people were on task only 50% of the time, which I think it's because also the task is pretty boring and pretty easy, so you don't have to pay attention a lot to have a decent performance at it. And then we had participants reporting mind wandering about 40% of the time, and about 10% of the time they were mind blanking. I will attract your attention to the huge variability between these participants. I mean, some participants were always on task, some participants were mind wandering all the time, some participants never mind blank, and other mind blank 50% of the time. So there is really a huge variability, which might be because there is actual variability. So maybe people have a tendency to mind blank more than others. It might also be to the fact that we are asking participants to categorize their thoughts in these three rigid states. But we're well aware that things are very dynamic, and it's not always clear cut. So you have also individual potential difference in deciding what kind of thoughts should be classified as on state or mind wandering, mind blanking. In terms of what happened across a block, so what you can see is something quite expected, that they start being pretty focused, and then this is decreasing within a block. what's interesting here is that toward the end of the block, they know it will be the last probe. So yeah, if they have, they seem to mind wander even more. And then across the task, so the task was composed of 6 blocks. You can see that you have a similar dynamic. So they start pretty well focused. And then the mind wandering increase, while the mind blanking is increasing as well, but fairly stable. And you have a last block effect where on state is increasing again because they know they're almost over. So which imply that, I mean, all of this is obviously modulated by motivation. So obviously, what we can do is to look at the behavior just before this interruption and to split the mind probes between the different states I reported, and to look at the behavior just before the participants were interrupted. And if you look at this, you can see that both mind wandering and mind blanking are characterized by a decreased performance, and especially a lot more false alarm. But what was interesting was to look at when looking at the reaction time, what we saw is that there is actually a difference between mind wandering and mind blanking. In the case of mind wandering, participants have the tendency to speed up, as if they were becoming more impulsive, while in mind wandering they tend to be more sluggish, so they slow down. So although the overall performance is decreasing, it seems that these still correspond to different ways of being distracted. We looked at participant vigilance before this probe, looking at both an objective marker of vigilance with pupil size. So the more you're awake, the bigger your pupil, but also asking participants to rate their vigilance at each probe from a scale from 1 to 5, 5 being very, very awake. And what you can see is that both mind wandering and mind blanking characterize low arousal state, which could potentially go against, for mind wandering, go against some results or models reported in the literature that have the tendency to associate mind wandering with a form of hyperarousability. So you're mind wander because you're too excited, too awake. This result, but others in the literature, seems to point out actually that when you're mind wandering, you're usually just sleepy. So we had here an interesting context for understanding this relationship between vigilance and attention, because we have a task where we have participants losing attention a lot in different ways, but all these different types of attentional lapses are associated with low vigilance. So the idea was to see whether we could capture the decrease of vigilance directly with the EEG, using a marker of sleep itself, and how that could explain not only behavior, but subjective experience. So to do that, we use this concept of local sleep, and we detected sleep-like slow wave in the scale PG. So slow wave that look like what you will observe in sleep, but local both in time and space. So there will be isolated waves in a subset of electrodes. So what we do to do that is we use the same algorithm that is used to detect sleep slow wave, which take the raw EEG, filter it in the slow oscillation bands. The only thing we have different from what is done in typical sleep studies is that we use a relative threshold to retain only the highest amplitude slow wave. So in sleep, you use an absolute threshold of 75 microvolts. We can't do that because we don't have such huge events in wakefulness. So what we do is we put the threshold and we take the top 10% of the highest amplitude slow wave per electrode. And what you obtain is a slow wave I showed you before that have a density, amplitude, and slope that is more prominent frontally, which is very typical of sleep slow wave. Importantly, they are distinct from task-related events. So I'm plotting here the ERP log to the stimulus or the response with their topographies, and you can see there's a slow wave that have on average they have a peak-to-peak amplitude here of 15 microvolt. They are 1 order of magnitude almost bigger than what you will see in terms of stimulus response stimulus ERP or response ERP, and the topographies as well are completely different. So we're not looking here at the classical ERP. This is really very high amplitude events occurring in the EEG. And what is interesting is when you construct mind wandering and on state, you can see that mind wandering is characterized by more of this slow wave, higher amplitude slow wave and sleeper slow wave. And this seems to be mostly true for frontal electrodes. So when you're thinking about something else, you have you can see more of this marker of sleep frontally. While when you're on mind blanking, although you have more of this slow wave frontally, their slopes, so how steep they are, which is a marker of synchronization of in the brain, is this time more over parietal areas. Mind blanking versus mind wandering contrast indicate actually that slow wave have a lower amplitude frontally in mind blanking compared to mind wandering, and they're steeper in the back of the brain. So it seems that not only the slow wave seems to increase in both mind wandering and mind blanking compared to when you're task focused, but the region in the brain where this occurs might be contrasted between these two attentional states. There is also a temporal relationship. So in the previous slide, we detected the slow wave in the 20 seconds before a probe, but you can do that with a finer time resolution. So going from 5 seconds to 5 seconds until the onset of the probe, and what you can see from this mind wandering versus on contrast is that the effect seems to be, I mean, not restricted, but larger in the few seconds that precede the probe onset, and this was maybe even more true for the mind blanking. So there is a tight temporal relationship between these events and what people report. But what was potentially even more interesting was to associate the presence of this slow wave with behavior, and this is you can do at the trial level, because for each trial you can look whether for the different electrodes you record you have or not a slow wave, and whether the presence of the slow wave for that trial and for a given electrode will be predictive of the behavior for that trial. So really you were working at the second level, and whether you can explain behavior with what happened during the trial itself. And when we did that, we saw a positive correlation between slow wave occurring in frontal areas and false alarms, which fits well with the function, the norm function of functional cortices in regulating behavior and executive control, and in allowing you to inhibit a response. You can imagine that if your frontal cortex is getting sleepy, so showing more of this slow wave, then you'll have less of an ability to inhibit your response. So maybe this positive correlation that we see here with frontal electrodes is just showing that. I mean, the prefrontal cortex are losing its ability to help you regulate your behavior. This topography contrasts a lot with the correlation we obtained for when trying to predict missed trial. Because this time we had a correlation mostly over occipital area, so potentially visual cortex. So this time you can imagine that if your visual cortex is getting tired and you're doing a visual experiment, then in the trials where you will have a local sleep event, then you're more likely to fail to encode the ongoing stimulus or to encode it poorly. That could lead to a miss. Here you can really see the power of these slow waves to predict behavior in a fine temporal scale, because we're working at the single trial level, but also with some region specificity, with slow wave having effects that are region dependent and depend on the function of the region we're looking at. And this contrast between frontal and parietal cortices can be found in the reaction time, where you see a negative correlation between the presence of slow wave and reaction time. So in frontal areas, so when you have a slow wave over the frontal electrodes, the participants have a tendency to speed up, which could explain the increase in false alarms. Whereas when the slow wave occur in the back of the brain, they have a tendency to slow down. this relationship between decision making and how people compute their response to the task was confirmed by doing a drift diffusion modeling, when you can extract for participants the variable of the decision and try to see if they are influenced by the presence of slow wave for specific electrons. And what we could see that the accumulation of the go response was slowed down. The drift for the go response is slower when you have slow wave, especially toward the back of the brain, whereas the frontal slow wave will slow down the accumulation for the no-go response, but will accelerate it when they're observing an occipital cortex. So again, this contrast between the effect of slow wave in the front and the back of the brain, resulting in a bias for go response that is decreased when you have occipital slow wave. So this result we're observing at the single trial level can be replicated through a modeling approach. So overall, what it seems we have here that slow wave that are able to predict behavior in a region specific way. And to predict different types of lapses. So when these slow waves occur in the front of the brain, they seem to be predictive of false alarms and mind wandering. When they appear in the back of the brain, them seems to predict misses and mind blanking. What is very interesting here is with one simple mechanism, so just a sleep slow wave occurring while you're awake, you can explain different cognitive consequences. Something important, although to note, is although we observe in this correlation map a contrast between front and back of the brain, something to remember with slow wave is that we know from the sleep literature that they travel, and they usually start in the front of the brain and then travel toward the back. And this seems to occur as well in waking. So if you detect a slow wave in the front of the brain, you have a quite high probability of detecting a slow wave as well in neighboring electrodes. But if you do that in the back of the brain, actually the slow wave you detect in the back of the brain are not restricted to the back of the brain, but they also engage the front of the brain. So although what we see is a frontal parietal contrast, it's also possible that the wave we observe in the back are just actually more widespread, and this is why they maybe are correlated with mind blanking, because it could be a more intense form of local sleep, leading to a more drastic consequence in terms of subjective experience. So you're not only missing out on the rest of the world, but also on your internal stream of consciousness. So I wanted to finish this talk by just showing some application of this work to other protocols that are ongoing. So this is work in progress. But something I'm particularly interested in is seeing whether this concept of local sleep could explain interindividual differences, and especially in attention. So I'm very interested in the attention deficit hyperactivity disorder, because the current models of ADHD, that ADHD could stem from a dysregulation of arousal. So this weird combination of sometimes not being attentive enough and sometimes being hyperactive could be due to the fact that ADHD individuals are oscillating between too low and too high arousal, and they can't maintain themselves in the optimum. What's also very interesting with ADHD that 50% of the adults with ADHD report sleep disturbances. So it's a population we know have trouble sleeping and maybe therefore have issues, vigilance issue during the day. And sleep deaths lead to actually very similar cognitive impairments to ADHD. So a combination of being too impulsive sometimes and being too slow at other times. So it will be very interesting to see if we actually could merge the two literature and explain part of the attentional disorder in ADHD with a vigilance issue and the intrusion of sleep during the day. So that's a bit my hypothesis. So if you're a well-rested, healthy, non-ADHD individual, during the night you will sleep well, and this means that all your brain regions will be able to rest well, which means that you'll be performing well during the day. Maybe what happens during ADHD, that you have sleep issue that makes part of your brain not sleeping so well, which will mean that this brain region will be sleepier during the day. We have preliminary data to back up this hypothesis, and we are recording more. The first one is a replication in ADHD control of the protocol I just presented. Another one is data I got from my colleague at Monash, Mark Belgrove, on a similar sustained attention task. The bottom line of this is that if you compare ADHD patients on their medication or off their medication to control, they show a lot more slow wave in waking compared to controls. So they're not just subjectively sleepier than controls. It seems that their EEG is also more sleep-like during the day than controls. Slow wave seems to positively correlate with the performance on the SRT. So the more they have slow wave, the more they will make errors in a sustained attention task. So it seems that the two things could go in hand, and potentially the slow wave are a mechanism explaining why they are not paying attention. We are trying to manipulate this local sleep intrusion as well with Mark Belgrove and the student of ours, Elaine Pingle, with pharmacology. And the idea is to, so it's data that, again, has been collected that we are reanalyzing. So people doing a different session of attention tasks while being given different drugs. And they in some sessions, so they have a placebo. Some sessions they have drugs that are stimulating, like methylphenidate, also known as Ritalin or atomoxetine, and they also receive the drug that is reducing vigilance, so citalopram. So these drugs have different effects on different neuromodulator. Methylphenidate is increasing noradrenaline and dopamine. Atomoxetine is more specific on noradrenaline, and citalopram is increasing serotonin. And what we are trying to do with this data is to understand how these drugs are impacting behavior and how they're impacting the EEG, and notably sleep-like slow wave, and whether the two could actually be understood together. So in terms of behavior, not very surprisingly, methylphenidate make people more better at the task, so shorter reaction time, less misses. Atomoxetine and citalopram increase reaction time, but citalopram increases misses alone, which fits well with the fact that citalopram is getting people more relaxed and also a bit sleepy. What was very interesting, and again, this is preliminary data, but we're progressing well on this data set, is that we could see an increase in the amount of slow waves, so this marker of sleep, only in the citalopram case. Which fits well with the notion that the only drug here that was taken by the participant that was promoting sleepiness. So maybe we can here show that specifically, I mean, some drugs will have different impacts on your behavior and so on. But when you decrease vigilance, what you have is an increase in the sleep intrusion, and that may explain why you have less attention and worse performance. And lastly, the presence of the slow wave was correlated with the behavior in this task. So we're able to replicate what we had in healthy adults by showing that the more slow wave you have in the front of the brain, the more you will have misses and variability in reaction times. So overall, the conclusion of this work is that it seems that we have a marker of sleep that's intrude in wakefulness that we can characterize at different levels. So from single units to whole brain recording, we can examine the consequences of these events on cognition, and in a complex way, in a region-dependent way. In the future, I will be very interested to see if sometimes this can be useful. I mean, why are we doing this local sleep? Is it because local sleep is some sort of a safe switch that our brain is using when part of the brain is feeling too tired. We switch it off to avoid the propagation of this tiredness to the whole brain, and to avoid cases like in narcolepsy where you just fall asleep abruptly without control. So is it sometimes useful to do local sleep? And as I explained you, I'm very interested in intervening, modulating the amount of slow wave using different approach. That can be drug, as I told you, but that could be also acoustic stimulation during sleep to try to improve sleep, to see if that will reduce the amount of sleep intrusion during the night. So thank you a lot for your attention. I think I'm, as usual, a bit over time, but we'll be very happy to answer your questions.